In the last class, we were talking about something very important. What was that? It was about muscles, correct? So let's go back there and we'll start from where we stopped. Okay. So while we were discussing about muscles, we were understanding how muscles work or how the muscular contraction happen. So while we were discussing that, we understood that muscles are arranged in the form of, in the muscle, if you take a cross section and observe. So the muscle fibers are arranged in the form of bundles, which we call it as fascicles. And within these fascicles, we have several muscle fibers. So that's what we discussed, right? So we understood that the muscle fiber has a plasma membrane, which we call it as sarcolemma, and then we have the sarcolemma enclosing this protoplasm of the muscle fiber, which we call it as sarcoplasm. Okay, so now I told you they're in multinucleated condition, and then the endoplasmic reticulum of the muscle fiber is called as sarcoplasmic reticulum. Very important things that we discussed. And there are also the calcium banks, okay, that store calcium and release calcium during the muscle contraction. Correct. So what we understood is while we look at a muscle fiber, and then while we are examining it, especially the skeletal muscle fibers, we see that there are dark and light alternating bands. So these dark and light alternating bands is because of parallelly arranged fibers or filaments called as myofilaments or myofibrils. So these myofibrils, as I said, they are arranged alternatively and parallelly. Okay. So the dark and light appearance of these bands is because of a specific nature of these protein filaments or proteins that are present within these myofibrils. So let's see. So this alternate dark and light bands that are present is because of, you see, myofibrils, light myofibrils such as actin and myosin. So what is actin? Actin is a light band, okay, and actin is also called as isotrophic band, okay, or I band we call it as. Whereas myosin is a dark band or a thick band, okay, which we call it as anisotropic band, and they contain myosin, obviously, okay. Now these proteins are arranged very parallel to each other and alternate to each other, okay, on a longitudinal axis. So now if you look at the what is actin filaments and what is myosin filaments, so let's see how they are arranged first. So if I take the isotropic band, when I talk about isotropic band, so that means I'm talking about who? Actin. 1 second, students, okay, I'm talking about who? Actin. So if I talk about actin, actin are called as I bands. So these I bands are parallel to each other, isn't it? So they are parallel to each other. So these I bands are not just independent, but they are held together by the elastic fiber. So there is an elastic fiber that stitches this light bands or isotropic bands throughout the length. So these I bands which are stitched together by this elastic fiber. So this line that holds them together is called as what line? Z line. It is called as what? Z line. So this is one. Okay. So this is one Z line. Let's say you have the fibers like this, and then not the same fibers. Let's say you have another set of fibers which are actin fibers, and then there is another Z line that is holding another set of actin fibers. So now you see this is one Z line and this is another Z line. So between these two Z lines is where the functional unit of muscle fiber lies. So when we talk about the functional unit of muscle fiber, we call it as sarcomere. Okay. What is the functional unit of muscle fiber? It is called as sarcomere. What is a sarcomere? Sarcomere is an area or it is a region between two successive Z lines. So whatever that lies between the two Z lines is called as what? Sarcomere. What is sarcomere? It is a functional unit of muscle fiber. So what did I tell you? There are parallelly arranged thin lines or thin filaments, which we call it as actin filaments. So these actin filaments, at one point, they are held together by these elastic fibers, okay, on a longitudinal axis. So this line that stitches all these actin fibers on a longitudinal axis is called as what? It is called as a Z line. Okay. So this is about actin. The same goes with the thick filaments as well. What happens with the thick filaments? These thick filaments are also parallelly arranged. Okay, they are also parallelly arranged, but it is held together by a very, very, very thin, okay, fibrous layer. So there is a small fibrous line that holds all this, myosin. Okay, which filaments? Anisotropic, that is A band. A band together are held together by a very thin fibrous membrane, which we call it as M line. So let's call this as M line. Usually it is so thin. Okay, so this is called as M line. All right. So we have Z line and we have M line. Who holds the Z line? What does Z line hold? It holds the isotropic bands or I bands or actin bands. Whereas when you look at in the between, in the center, what do we have? We have thick filaments, and these thick filaments are called as myosin filaments. And these myosin filaments are held together by a very, very thin. See, remember, the Z line is an elastic line, elastic band, whereas the M line is a very thin fibrous membrane. Okay, it is a fibrous membrane. Now, as I told you, these both isotropic or and anisotropic bands are arranged throughout the length of the myofibrils. Now, what happens? Let's see one more fact about it. One second. Okay, when you look at the muscle in a resting state, that means when they are not contracting or they're in a very relaxed state, let's say. So while you're sleeping, that's the best way. So or the muscles in your arms or muscles in your legs, how do they appear when you look at it? So when you look at it in the resting state, what you see is there is an overlap of A band over the I band. See, you see this is the you can see that this is the I band. The I band overlaps over the A band, correct, right? It overlaps throughout the length of the A band. So what is A band? Your anisotropic band or your myosin. So, okay, let me keep it very simple. When I say myosin, just think that I'm talking about A band. When I say actin, think that I'm talking about I band. Okay, students. So what happens? They uh, overlap. Overlap means what? See, this is the A band, and then this is the I band. So the I band is not supposed to overlap while it is contracting. What it does is it overlaps in the resting state. Till where does it overlap? It overlaps throughout the length apart from the center. In the center, they do not overlap. So in the resting state. So this is called as head zone. What is an head zone? In the resting state, okay, the I band overlaps the A band throughout the length except at the center. So this non-overlapping region is called as what? The head zone. Okay, very, very important. So you understood what is head zone, right? So if they ask in the resting state, so the central portion of a sarcomere is made up of what? The central portion of a sarcomere in the resting state is made up of only which band? A band. Okay, it does not contain any of the I bands, especially in the resting state. Okay, students, so this is about head zone. Now let's talk about the structure of contractile proteins. First we talk about actin. You see, uh, uh, in a regular cell, not the muscle cells I'm talking about, even our regular cells have actin monomers that help in forming the filaments of a cytoskeleton. Okay, but here, how does the actin formation takes place? If you look at this actin, actin is a helical structure. Helical structure means like a staircase-like structure that is arranged. But before it became a helical filament, okay, it is just one small bond. So that means there were monomers for this actins. Okay, so this actin monomers, okay, so these actin monomers are called as globular actin monomers or monomeric Gs. Okay, so these monomeric Gs on activation, they come together. So when they come together, they become F-actin polymer. Okay, so when they become F-actin polymer, they arrange themselves in such a manner that they are wind together in a helical fashion. Understood? So once they are held together in a helical fashion, what happens? The most important part is the way actin becomes useful is when it gets a place to interact with something called as myosin. Correct, right? So the thick filaments and the thin filaments have to interact at one point. So this interaction does not happen with just effactins or the effactin helical structure. It happens with the association of a specialized complex protein. So the specialized complex protein is called as tropomyosin. So what does this tropomyosin do? The tropomyosin will run throughout the length of the actin. Okay, it runs throughout the length of the actin. Okay, having an association with your actin monomers. So there is an association of actin and tropomyosin. Okay, okay, troponin and tropomyosin throughout the length. So this is about the actin. Now what happens is, here and there, okay, at a periodic intervals, let's call it as at periodic intervals for each helical round, or that means for each turn, at every turn there is a special association of another protein, which we call it as troponin. So what happens is, the troponin has very good affinity. Affinity means a friendship, a bonding, an attraction, you can call it as. So what happens, especially in the resting state, especially when we don't use the muscles? So this troponin makes a contact, makes an association with the tropomyosin, so making it troponin tropomyosin complex. So when the troponin tropomyosin complex is formed, that means there are no active sites, or there are no possible binding sites for our thick filaments to interact. Let me repeat. So when we look at the structure of actin, actin looks like a double chained filamentous structure, okay, helical filamentous structure, and each of the actin filament is made up of monomers of actin, which you call it as globular actin or G-actin. So this globular actin, upon activation, they come together and they wind together in a helical form, creating this actin filament. Throughout this helical chain of actin filaments, there runs, okay, a continuous chain of very complex protein called as tropomyosin. So the tropomyosin winds around this actin. So now what happens is, the tropomyosin is one thing that gives the place for this thick filaments to interact. Okay, but what happens during the resting state? There is another protein that is interacting, which you call it as troponin. So the troponin, which is periodically placed, so this troponin creates a complex with tropomyosin. So there, what it becomes, troponin tropomyosin complex is formed at what position? Whenever we are not using our muscles. So that is when troponin tropomyosin complex is formed. So this is the structure of our um, actin. Okay, so let's go for myosin. Okay, myosin, it looks like a snake basically. So myosin is also made up of monomers. Okay, it is also a polymer protein. It is made up of monomers called as meromyosins. So this meromyosins, they make up the thick filament, one thick filament. Okay, so this is one meromyosin. Okay, this is one meromyosin, and this is another meromyosin. So there are two meromyosin, which makes it a polymer. Okay, so now when you look at the meromyosin, it has two important part. One is the head part, other one is the tail part. Usually when you look at this region of the head part and the tail part, there is also another region that is called as a neck region. So the neck region plays a very important role. Why it plays an important role, we'll talk about it. So when we look at the meromyosin or the myosin, it has two regions. One is the helical tail region, and then a projected head region with the support of a neck. Okay, so Now, when we talk about this myosin, we can divide it into two important components. What are those? If you look at one region, it is made up of heavy regions, and the another region is the light region. So we can call it as heavy meromyosin, HMM, heavy meromyosin, and then we can call it as LMM, that is light meromyosin. Light meromyosin is very less dense, and heavy meromyosin is high density. Correct, right? So now, if I talk about the light meromyosin and heavy meromyosin, they are very, very important. Why? Because if you look at the light meromyosin, they form this helical tail, okay, that supports your myosin. And then coming out of this tail is what is a projected neck. So the neck is placed in such a way, okay, it is we can also call it as a short arm. So it projects outward at a very regular interval, okay, so that your myosin head can create a cross arm, okay. Now, if you talk about the globular head portion is highly important happens, okay. So sorry. So when we look at this globular head, there are two heads, okay. One is like this and the other one is like this. So one head is known for its affinity for the ATP, okay. It goes and attaches to the ATP, and the other head has the binding site for actin, okay. So the head of heavy meromyosin or the myosin has two important functions. So one head is having a binding site or an affinity site for your ATP molecules, and it also has the capacity to hydrolyze your ATP into ADP and phosphate. Okay, it binds the ATP, it collects the ATP, it breaks the ATP, and when it breaks the ATP, it converts ATP into ADP and phosphate. So what happens? The energy is released. So this energy is used for some purpose. Okay. And the second one is I told you that these thick filaments and thin filaments have to interact at one point. So this interaction comes with a binding site. Everything is a binding site. Or they have to have a point of attachment. So this point of attachment to the thin filaments or the actin comes with the another head. So this is called as actin heads or actin binding regions. Understood? So the two filaments, two heads. One head is for ATP binding site and the other head is for actin binding site. Are you following me, students? Did you understand this? The structure of myosin. What did I tell about structure of myosin? A myosin is also called as a thick filament. It is a polymerized structure, and the monomeric unit of the myosin is called as meromyosin. So the meromyosin is of two parts. One is light meromyosin and heavy meromyosin. The light meromyosin is responsible for the formation of the tail of the myosin, whereas the heavy meromyosin is responsible for the formation of head region of the myosin. So now the myosin, apart from the light tail and a heavy head, the heavy head has two important region. One is the ATP binding site is present, and the other one is you have the actin binding site is present. So this is very important facts that you have to understand with respect to myosin. Is it clear? So if the question is asked, what is the monomeric unit of your myosin? You have to tell it is meromyosin. Okay. And then what are the different types of meromyosin you have? You should tell it is light meromyosin and heavy meromyosin. Okay. Is it clear? Do you guys have any doubts? Let me know. Are you following? Are you understanding, students? Pradeep and students from Ramnagar? Yes, no? Okay. Who is that? Okay. Jainvi Ramnagar students. Chalo. So I will continue. Okay. So now, how does the muscle contraction happens, or how does muscles work? So the mechanism of muscle contraction can be explained through something called as muscle contraction theory. It is also called as sliding filament theory. There is a reason why we call it as sliding filament theory. Okay, I need a diagram. Let's see, we have a diagram. Okay, we have, excuse me. Okay, no problem. See what happens. First of all, it all starts with a stimulation. Okay, there has to be a stimulation. Give me a second. Uh, an empty page. Oh, sorry, just one second. One second. Um, Okay, let it be here. Okay, first it starts with a stimulation. First, you need to have a thought process because it is obviously a skeletal muscle. Without your command, your muscles won't contract. So you create a thought process, right? So and then your thoughts or your stimuli is taken out by a motor neuron. The motor neuron carries this signal, but what happens is these neurons have an end plate. Okay, so let's say that this is your um, the synaptomial end. Okay, and then you have your muscle tissue. So this is your muscle tissue. Okay, let's say. So now what happens is the nerves do not directly and see, you cannot think like it is an electric circuit. Okay, see, this is the muscles, this is the end of your neuron, and this is the muscle. So they can only reach up to a certain point, and they don't directly go and physically control the action of these muscles. So that transmission of this signal, see, nerves are carrying some signal and it has to deliver it to the muscles. So the delivery of the message or the signal that these motor neurons carry can reach up to the motor end plate. That is at last you can reach your nerve system. So what happens after the motor end plate? Now what happens is the signals from the neurons are transmitted through what do you call? Mm, um, messengers, or we also call it as neurotransmitters. Okay, they are transmitted through neurotransmitters. So these neurotransmitters, what they do is they are released into this space or the synapse. So when they are released into the synapse, so these neurotransmitters, they go and bind to the receptors that are present on the plasma membrane or sarcolemma, right? So the plasma membrane of the muscle fiber is nothing but sarcolemma. So once they go and bind to the sarcolemma of the muscle fibers, what happens is it creates a rapid depolarization. Okay, what is depolarization? The electrical conductivity of this muscle fiber completely changes. So it starts, let's say that this is your surface of the muscle fiber and this is the receptor, and the which one, a neurotransmitter is coming from the nerve plate. Okay, and they release this neurotransmitter, and let's call this neurotransmitter as acetyl acetylcholine or acetyl CoA, which is a common neurotransmitter, usually not CoA, it is choline, acetylcholine. So this acetylcholine is a most common neurotransmitter. It's like a messenger. So now what it does, from here it goes and binds to this receptor. Once it goes and binds to the receptors, the conformational changes, what it will do, it will create immediate depolarization. So the immediate depolarization of the sarcolemma or the plasma membrane, what happens is it will activate or it will also depolarize which one? Sarcoplasmic reticulum. Which one? Sarcoplasmic reticulum. So when the sarcoplasmic reticulum is activated, what do sarcoplasmic reticulum have? I told you the endoplasmic reticulum of the muscles is also called as sarcoplasmic reticulum, and they act as calcium banks. So now what they do is they rapidly release this calcium ions. So when the calcium ions are released, let's get back here. When the calcium ions are released, so these calcium ions that are released by your sarcoplasm, what happens is they go and bind to this troponin. So once they go and bind to this troponin, so now in the resting state, what was happening? You had tropomyosin, and to this tropomyosin, troponin was coming and binding, creating what? Troponin tropomyosin complex. So when the troponin tropomyosin complex is formed, now there are no myosin binding sites. That means myosin cannot find any place to interact with actin. So that is why it was not possible for any contraction. Now, since the calcium is released, so these calcium ions go and bind to the troponin. Once they go and bind to the troponin, it creates conformational changes in troponin. Okay, which changes? Troponin, troponin changes. When I say conformational changes, there is a small structural changes that happens in troponin. Now, troponin can no more recognize or no more can it attach to the tropomyosin. Since there is a conformational change, this troponin tropomyosin complex is broken. Once this troponin tropomyosin complex is broken, now what happens here is that tropomyosin, okay, this becomes, okay, there is no troponin anymore, so this becomes active myosin, myosin binding site. So it becomes active myosin binding site. When it becomes an active myosin binding site, now the door is open for which one? For your myosin head to interact with the actin. Correct, right? So now what happens? Now first, we wanted a stimuli. So we wanted to contract our muscles. So we send this signal through a motor neuron. The motor neuron took this messages in the form of impulse and it traveled all the way to the edge of the neuron. And this edge of the neuron is called as motor end plate. So once it reached the motor end plate, it wanted to transfer this message to your muscle fibers. So it cannot do it. So it sends the message through a neurotransmitter, which you call it as acetylcholine. So the acetylcholine, having the once the acetylcholine is released into this neuromuscular junction or NMJ, we call it as, this junction is called as NMJ or neuromuscular junction. That is a sign that muscle needs to be contracted. So this acetylcholine goes and binds to the receptors of the sarcolemma. So that will create a huge depolarization. So once this depolarization rapidly spreads across the rest of the plasma membrane or sarcolemma, that will activate your sarcoplasmic reticulum. Sarcoplasmic reticulum released which one? Calcium ions. So these calcium ions, they go and bind to this troponin, breaking the troponin tropomyosin complex on the actin. Okay. So once the actin filaments are free of troponin, now this will create a site. What site? Active binding sites for myosin. So once the active binding sites for myosin is created, now what happens next? Stage two. In the stage two, what makes these myosin heads react? That is the biggest question. So the myosin, how does it know that okay, now the gate is open, now I can just go and attach? That doesn't happen. So now an ATP molecule plays an important role. This ATP molecule comes and bind to the ATP binding site. Okay, ATP binding site on the myosin head. We have two heads, right? One head for ATP binding site, another head for actin binding site. So on the ATP binding site, one ATP molecules comes and attaches here. When the ATP molecule comes and attaches here, suddenly it's like an attraction. All of a sudden, this myosin gets so attracted, so excited that it increases its affinity. Okay, it increases its affinity to go and attach to the actin. Now, the myosin was in a very resting state. Okay, once the ATP bound, now it becomes very affined attracted to the actins that are here. Let's say that this is the actin that we have. Okay, let me erase this. Okay, let's say that this is the actin that we have. And to move towards the actin, okay, myosin required which one? ATP. So once ATP binds, it gains the affinity. Now, to raise its head and bind to the active sites, what does it need? It requires energy. That's when it hydrolyzes the ATP. It breaks down the ATP into ADP plus phosphate. So once this breaks the ATP into ADP plus phosphate, now it gains the energy. Now what happens? This myosin, using its neck, it gets straightened up and it binds to the active sites on the actin. So this actin, this is myosin, and ATP is used, ATP is hydrolyzed, it becomes ADP and phosphate. Now, when you have this actin filament, and then when you have a myosin head, the myosin head raised, and then it attached to the actin on the active site, creating actin myosin complex. So this actin myosin complex, okay, is also called as cross bridge. Cross bridge. Okay. Now this cross bridge is created, and your myosin head is in the cocked position like a gun. Okay, it's in the cocked position. Now, once you have a cross bridge formation, now the bridge has to be pulled backward. You see, it has to push this actin forward, and the head has to stroke backward. So, does it happen without energy? No. Again, what happens? Now, this is the actin. Now, this is the myosin tail, and this is the head. Okay. Now, the cross bridge formation has taken place, something like this. Now, let's say that this is the myosin binding site. This is the ATP binding site of the myosin. Cross bridges happened. Now the ATP binding site is there. One ATP molecule we already hydrolyzed for raising the head. Now to create the stroke, okay, what is the stroke? The myosin has to break the cross bridge, move backward, push the actin forward. So when this has to happen, another ATP molecule comes and binds to the ATP binding region. Here it immediately hydrolyzes. Using this tremendous energy, it will stroke back. Okay. Once it strokes back, it pushes the actin forward, and then the myosin comes to the resting state. Is it clear? Now what happens? Again, one more ATP comes, the head raises, the cross bridge formation takes place, and the ATP comes, hydrolyze, gain energy, break the stroke, okay, and then move the actin, making these fibers contract, okay, towards the center. So once this contraction happens, keeps happening towards the center, you have the muscle contraction. Okay. So did you guys understand this? Yeah. Easy, right? It's not that difficult, but it's like a story. Okay. Is it understood? All right. So let's go for the next one. Okay. So we understood how the cross bridge formation takes place and how the ATP is synthesized. Okay. And how your muscle contraction happens. Remember, until and unless we have ATP, okay, and calcium ions, we can have the muscle contraction. Once you don't have any more energy, the cross bridge formation cannot take place. When the cross bridge formation cannot take place, you cannot have muscle contraction. That is why ATP becomes a very integral part for the muscle contraction to happen. Now sometimes what happens is, when you're coming back in the relaxation state, when you come back in the relaxation state and there is no stimulation of the muscles anymore, now the calcium ions, okay, are pumped back to your sarcoplasmic reticulum. Once they come back to the sarcoplasmic reticulum, now this marks that actin filaments can no more interact with your myosin. So once there are no calcium ions, the calcium cannot bind to the tropomyosin. Now the troponin comes back and occupies its original position, creating troponin-tropomyosin complex. Okay. Now myosin don't have any binding site, so myosin comes into resting phase where your muscles will start relaxing. Okay. So this is about relaxation and contraction. Now you can see here, sometimes what happens is, you need continuous supply of oxygen for your tissues. So the continuous supply of oxygen to the tissues ensures that there is sufficient amount of ATP present for this muscle contraction to happen. But since we work hard too much and then we keep on contracting and our breath is going too rapid and we are not able to supply sufficient oxygen to our tissues, at this point what happens is our tissue actually goes into anaerobic respiration, where the lactic acid accumulation takes place. When the lactic acid accumulation takes place, that is when you have muscular cramps, okay, or muscle cramps will happen. So muscle cramps is due to the accumulation of lactic acid first thing, and your muscles will be craving for more and more of ATP. Okay, and for that, your muscles need to break down lots of glucose. To break down lots of glucose, you need lots of oxygen. So when this is not compensated, that is when your muscles will go and arrest into cramps. Maybe all the sports guys would have experienced cramps once or twice in your life, or even when you do excessive work like shifting your home, or when your mom or dad gives you work in a farm, or when you're given a lot of work by your physical education teacher as a punishment. they would have experienced cramps. Okay, so next one is we can divide this muscle tissues into two important categories based on the presence of a specific pigment. I'm not talking about hemoglobin, but we call it as myoglobin. If there is myoglobin, your muscles will start appearing pink or reddish. So this myoglobin is a specialized pigment that carries oxygen to your tissues. So they highly depend on the presence of oxygen. Okay, so that is why they're involved in aerobic fibers. They are also called as aerobic fibers. Which one? Muscles that are high in myoglobin. Some muscles are very low in myoglobin. We call it as white fibers or white muscle fibers or white muscles. So these white muscle fibers, they have very less mitochondria. Okay, and they have very high sarcoplasmic reticulum, and they depend on anaerobic process for energy. So here they depend on aerobic process for energy. Here they depend on anaerobic process for energy. Here the number of mitochondria is very, very high. Here the number of mitochondria is very less, but sarcoplasmic reticulum is very, very high. So this is a very important difference that you have to understand when it comes to muscles. Okay. Now, so as I told you, whenever you have repeated activation of these muscles, they can lead to accumulation of lactic acid due to anaerobic breakdown of glycogen into lactic acid. That can cause you cramps or muscle fatigue, what we call it as. Okay, chalo. Next, we have to go for skeletal system. Before we go for skeletal system, did you understand this? What is thought? Hmm? All right. So let's go and discuss skeletal system. So when we talk about human skeletal system, it's a very important part for our body. That's what gives you your structural integrity. That's what makes your body rigid. That's what gives you mobility. That's what helps you to chew, talk like me. Okay, not like me, but everybody else, right? So muscles, how they're important for muscles to have a specific shape, structure, and function. Okay, we require the help of bones as well. So first, let's see. It's a very simpler exercise to understand our skeletal system. So our skeletal system basically is composed of bones as well as cartilages. So bones are very, very hard, as you can see, whereas cartilage is quite flexible. It's quite elastic. You can move and bend the cartilage. It does not break like bone. If you start bending your bones in your arms or legs, it will definitely break, isn't it? So why the reason is you have your bone cells, right? Let's say you have your bone cells packed. The bone cells are placed here and there. Between these bone cells is what we call it as the matrix. So the matrix of the bone is made up of uh, very hard salts, which we call it as calcium salts, such as calcium pectate or calcium carbonate, and as well as minerals. That's why the bone is very, very hard. Okay, and then you have cartilage on the other hand. The ground matrix of the cartilage is made up of chondroitin salts. So this chondroitin salts is also calcium derived, but the composition of these salts make this cartilage very flexible. Okay, not as rigid as bones. Whereas coming to the bones, they make up all the major components of our skeletal system. So we have around 206 bones in our body, and this 206 bones in our body can be categorized into two important categories. One category of bones we call it as axial skeletal system. They make up axial skeletal system. Another component of it makes up to appendicular skeletal system. So we have axial skeletal system and we have appendicular skeletal system. So when it comes to axial skeletal system, what is axis? Axis is something that is in the center, let's assume. So we have what is in the center that is attached to your centric portion of your body. So that includes your skull, that includes your vertebral column, that includes your ribs. Okay. So the skull, the vertebral column, and the ribs, they are made up of axial skeletal system. So there are how many bones in axial skeletal system? 80 bones that are present in axial skeletal system. Whereas when it comes to appendicular skeletal system, your forelimbs and hind limbs, they make up to appendicular skeletal system. So your appendicular skeletal system, they comprise of 126 bones. Okay, so it's not possible to do any of this activity without the help of bones, isn't it? So let's see axial skeletal system. As I said, the axial skeletal system includes the skull, the vertebral column, as well as the rib cage, okay, or the thoracic cage. So let's see what are the facts about these axial and appendicular, sorry, axial and appendicular skeletal system. It's not moving. So when we talk about the skull, or the skull is also called as the crania or the brain box. So the brain box is not just one bone, it is a composition of many bones. So our skull includes two sets of bones. One bone is the cranial bones, that it is also called as the brain box, and the other one is the facial bones. So in total, the cranial and the facial bones is 22 in number. So the total bones in the skull is around 22 in. So if you talk about cranial bones, there are 8 in number, very important. They're all stitched together. Okay, so we call it as cranial fascicles. Okay, or features. So these cranial features are like stitchings, which are made up of fibrocartilaginous tissue, which makes it immobile and very very tough connection between these plates. Okay, they're all flat bones. Okay, now when you talk about this crania, you can see that some of the examples of this cranial bone is sphenoid bones, frontal bones, temporal bones, parietal bones, occipital bones. Okay, so they are the examples of your crania. You might think that why you marked only 5. Now, if you see temporal bone, there are 2 in number. Similarly, the frontal bone is 1 in number. Okay, the occipital is 2 in number. So they totally make up to 8 in number. Okay, so when it comes to facial bones, the facial bones is made up of 14 skeletal elements in the frontal portion of the skull itself. And then when you look at your entire skull, you can see that the only movable bone is your lower jaw, and the lower jaw is called as the mandible or mandibular bone. So this mandibular bone is the only movable bone in your skull. Now, there is a U-shaped bone in the basement of your skull, which is called as a hyoid bone. Okay, so that is present in the base of the buccal cavity. Okay, even the hyoid bone is a part of the skull, even though it is not completely attached to the human skull. And also, if you look at your cranial portion or if your skull region, what you can see is it is even our ears, it has 3 important bones in each ear. So they are malleus, incus, and stapes, which we talk about these 3 bones very detailedly in neural control and coordination. No problem. So which we in general we call them as ear ossicles. So each side of the ear has 3 3. So 3 plus 3 is 6 bones. Okay, so if you see the skull is having a very good association with the vertebral column. That means it articulates or it is very nicely attached with the skull. So this articulation means attachment of the skull with the vertebral column, okay, happens with 2 occipital condylites. Okay, so we call it as occipital condylites at the base of the skull. You can see here. Hold on. So the base of the skull here, we have something called as occipital condylites. So this occipital condylites, so here occipital bone. So there are 2 occipital condylites like this. So that is why we call it as dicondylitic skull. So the dicondylitic skull means there are 2 occipital condylites that helps in the attachment of our skull with the vertebral column. You have to remember the vertebral column is attached to the skull. How? Through what? It is attached through occipital. The human skull is what type? We have dicondylitic skull. Why? Because we have 2 condylites. Okay. Chalo. 1 second. All right. I think we'll stop here because the vertebral column is again a new topic and another 10 question. Then we can start with neural control and coordination, and hopefully we can finish it on time. Now the session is open for your doubts. If you have any doubts, you can ask me. Okay. Please remember the number of bones. Okay.